very much, Toppin, for that very kind introduction, and thanks to the organizers and to the CLICK program for inviting me and my colleagues to come and experience a bit of Singapore culture, to understand the ambitions in Singapore to promote lifelong learning, providing new tools for children to do better in school, and also how that can apply as we age to provide a healthier lifestyle. Now, my talk today, as Toppin mentioned, is on a substance called myelin. And if you've never heard of it before, I'm hoping to give you a sense for why this substance is so important. And it came into the evolutionary framework as the brain became more complex and turns out to be critical for the way that we think and the way that we learn. So that's the kind of topic of this conference. And why do I say trapped in the brain without myelin? I'll try to give you a perspective on a pediatric disease in which there's no myelin in the brain and how devastating that is for children that are affected, but also how there are new therapies coming around the corner that may offer real hope for children like affected by this condition. So what I'd like to cover today is building a connected brain. We heard from Professor Trevor Robbins this morning about connections between the brain that we can measure by MRI, using a sort of magnetic resonance imaging approach to look at how parts of our brain are connected. And we can see that in a non-invasive way, but then also there have to be cells that are connected to each other directly. And I'll talk about how we build that up and how myelin is essential for ensuring the effectiveness of those connections. I'll talk about the structure and function of myelin, its role in evolution to build a more complex brain, how it can play a role in learning, and also diseases of myelin and therapies of the future. And as Toppin mentioned, I've had a foot in the lab and also in the clinic. So I'd also like to try to give you a dual perspective. And for some of you who may be thinking about careers in medicine or biomedical research, how it is possible to both work in the lab, but also translate those ideas. And by translate, we mean taking them into the clinic and seeing them through to potential benefits for patients. So I'll try to give you a sense of that dual perspective. Okay, so let's start off with the brain. You know, I think we heard from the CEO of NRF this morning that he wanted to learn more about the brain stuff. So what is the brain kind of made of? It is a complex structure, but there are primarily three major cell types, and they come from what we call neural stem cells. These are found in particular parts of the brain, and they're cells that are rapidly dividing to make more cells, but then they also spin off progeny. And as they spin off these progeny cells, they start to adopt particular types of characteristics, and we would broadly call them neurons or glia. And the three types are shown on the bottom. The blue is the neuron. It has axons, and these axons are the way that cell will connect to the muscle or connect to other nerve cells. And these are forming the connections that form neural networks. The red cell is the oligodendrocyte. That's really the star of this performance. And I'll talk a lot about what the oligodendrocyte does. That's a cell that will make myelin. And on the right is the astrocyte, which is also critically important for neurological function, but I'm afraid it doesn't get very much coverage in this talk. So now these videos will give you a sense of maybe, you know, activity that's happening in our brains, where as you sort of see the video honing in on particular nerves, you'll see the axons, you'll see electrical impulses moving along the axons, and then you're thinking to yourself, well, what are those little white blobs? Covering the axon, these will be segments of myelin. And so you can think of an axon as an electrical wire, and most electrical wires will need to have insulation, otherwise you get a short circuit. And that's very much what myelin is doing. It's allowing for insulation of that nervous impulse in the axon, but as I'll tell you, it's also enhancing the speed of conduction. The bottom shows a little bit higher detail where you can see now cells sort of connecting to each other through these myelinated segments. And these are found primarily in the white matter of the brain. And we have different ways of measuring white matter. And so this is one of the techniques that I'll be referring to. And also we heard about this in Professor Robbins' talk earlier, magnetic resonance imaging. I'm sure most of you are familiar with it. You've probably had, maybe you've had your knee checked, or maybe you've even had a brain scan by MRI. But what this is showing you is how we can measure the tracts in the brain that are forming connections. In this case, you see different colors, and those are what we call pseudocolors. They're just highlighting different tracts that are connecting the front of the brain to the back of the brain, or you may see things that look like they're crossing from left to right, and those will be going to interconnect the hemispheres. We can measure that by MRI, so it's a fantastic technique to understand the white matter. We can do this in patients that are alive, and that's, a real benefit for the study of myelin and how it can be affected in disease. So why do we call it white matter? Don't have to use your imagination. If you had a slice of the brain, this is a slice of a human brain, you can see why we call it white matter. And you might ask the question, why is it called gray matter? Maybe it should be called pink matter. But the white matter is where the axons that are coated with myelin live. They're white because myelin is a very lipid-laden substance. It is white in color, and so that's what gives the color to the white matter. Now, if we also wanted to look in more detail, I showed you that picture of the MRI, but then the one on the bottom shows what the actual bundles of axons look like, and you can see how nicely MRI captures that, because you can see those bundles, in this case, crossing from left to right hemisphere, and how the MRI gives you that sense of those sort of like cords of different axons moving together. So those are some of the techniques that we can use to analyze white matter at the gross level, or at a low resolution level. Now let's hone in more and let's really see the work that this oligodendrocyte has to do. This is the myelinating cell that's providing these blobs. So oligodendrocyte is sitting in a bundle of different axons. In this case, the axons are colored orange, and you can see that it has these arms like an octopus reaching out, enwrapping the axon in this elaborate bundle, and this is what we would call the myelin sheath. And then notice it leaves a gap. And this gap is called the node of Ranvier. It's a really important thing that you see with myelin, which is there's a lot of ion channels that collect in these nodes of Ranvier. And then you have the myelin in a segment, like a white blob, a node, another segment starts. And what the significance of that is that this is how myelin starts to help our nerves to work faster. Let's look at another non-myelinated creature, giant squid. Don't want to meet this probably if you're swimming. But how complex is the central nervous system of a giant squid? And this sort of shows the nerve cells of a squid. There are very few ganglia, and it's a very simple organism, has very low brain complexity. And also its axons, you almost can get a sense for how big they are compared to the animal. They're massive. And so what we see in non-myelinated animals like the giant squid will be axons that are very large, and they can achieve a speed of 25 meters per second. That's faster than any of us can run, but in terms of nerve velocity, it's relatively slow compared to axons that are myelinated, which are now 120 meters per second. And now you can start to appreciate where advantages for predation or escaping a predator come in. But also notice how much smaller the diameter of the myelinated axon is. It's a fraction of the size of these large diameter non-myelinated axons. And now if you think that every single axon is connected to a neuron and the axons are smaller, you can pack more of them into a single space, more axons, more neurons, greater brain complexity. And that's reflected in the numbers of neurons that we have, 10 to the 9th to 10 to the 11th, compared to the number of neurons Mr. Squid has, 10 to the 5th. And also you can see we have a larger brain proportionate to the squid. We're 2% brain, and the squid is 0.75% brain. So myelin has allowed that packing of more efficiently of smaller diameter axons that gives us evidence, that gives us some tools for creating brain complexity. Here's a paper for any of you that are familiar with the science literature. This is from the journal Nature, one of our kind of high-profile, high-impact journals. And it was looking in a kind of non-biased way at what are the differentiating features of a human brain compared to our non-human primate related organisms, let's say like gorilla or chimpanzee. And one of the cells that turned out to be different was from the oligodendrocyte lineage, again suggesting that there may be something in the transition from non-human primates to humans that also provides some focus on the oligodendrocyte. So myelin, it's an insulating coating of the nerve axons. It enables faster nerve conduction. packing of larger numbers of axons in a given space and has enabled, therefore, greater brain complexity. Okay, so I've taken you through the first 3 points. We're doing pretty well. Now let's talk about myelin's relevance for learning. So we wanted to bring this topic in because of the focus of CLIC, which is to really think about ways that we can enhance learning, and is one of the mechanisms that we should think about to potentially play a role in our learning abilities. And I'm going to focus on a couple studies from Bill Richardson's lab. He's based at University College London, but also other studies that are highlighted here in these references using MRI to monitor how the white matter changes in the context of learning. So let's imagine now that you're not a musician and you're given a violin and you're just starting to learn to play the violin. Steep learning curve, of course. But if we were able to measure your brain by MRI and focus on the white matter of your brain before, during, and after your learning, you could start to see changes in white matter structure, and those can be seen within 2 hours of learning a novel task, suggesting that the nerves and axons really are activated in the context of learning. That is reflected somehow in changes in the oligodendrocytes that are then showing signs that we can even see by MRI. So it's that kind of information that got Bill Richardson very curious. He's really one of the global experts on the oligodendrocyte. And so he asked the question, is myelin needed for learning? Now, I'm going to take you through the test that he used and maybe give you the perspective of how a mouse might have to learn. And this is using a test that's called the complex wheel. And so this is a video which shows a mouse on a complex wheel. Now, a complex wheel, mice like to run on wheels, but the complex wheel lacks rungs in unpredictable patterns. So you can start to see what does this mouse do? Now, this is a really curious mouse because he's trying to run, but he has to figure out where all the gaps are. He can't really run at speed. And so you can see him, he's making some mistakes. But believe it or not, he's remembering all of this. And this is a mouse in the active phase of learning. Now you could then say, well, maybe his myelin is changing. And now he's much better. That's after 1 week. He can go at speed, and that's the mouse that's learned to task. So that's how you can actually monitor complex motor tasks. And so just to give you a sense of how the scientists would measure that. Now this is the data from a paper from Bill in which he compared a normal mouse, which had that behavior I just showed you in the last slide. If you follow them for a week, here's the blue normal mouse. They really can get to speed pretty nicely after a week. Then he's compared that to a mouse that lacks the ability to make myelin. And what you can see is that if you prevent the formation of myelin, the mouse really doesn't have the same ability to learn the task. And this was the first evidence really showing how myelin was a critical substance that could enhance learning. And is now spawning, I think, a number of other studies. They're examining the role of myelin, both for cognitive tasks and also complex motor learning. Now, there are other reasons that we should also just think about myelin, let's say, give myelin some respect. It does change over time. And I'll show you sort of developmental changes in myelin that can also be reflected in behaviors over human development. But one of the times when there's a lot of changes, and actually can be very challenging sometimes in school, is during adolescence. And during adolescence, there are really a lot of changes in the brain. And that can be reflected in MRI, and that can be quantified sort of by measuring the volumes of gray matter and the volumes of white matter. And this study shows changes in the gray matter in boys that are in blue and females that are in red. Another that you might be familiar with is multiple sclerosis, which is the most common cause of neurological disease in young adults. That's an autoimmune disorder in which the body will attack the oligodendrocyte and cause depletion of white matter and neurological problems. And the one I'm going to talk about, you've never heard of, I'm sure. Well, maybe some of you are pediatricians, so you've heard of Pelizaeus-Merzbacher disease, or PMD. And this is a disease in which myelin is completely lacking. Okay, so let's again maybe talk about the developmental aspects. When do you start to see myelin in human development? And maybe ask the question, does the baby have myelin in its brain. And here's an answer. It's from a classic study done in 1920 where this investigator, Fleshig, used a stain for myelin, taking samples of human brain from 30 weeks gestation. Of course, this will be a preterm baby because gestation is about 40 weeks. So this baby, unfortunately, born early and sadly died before birth. A term baby, 4 months of age, 2 years of age. So you can see with the black staining when the myelin starts to emerge. In the preterm baby, there's virtually none. In the term baby, you see some in the brainstem, but not the cortex. Well, the brainstem is also where our breathing control is, and also a lot of the motor behavior for feeding is located. So that's pretty smart. Baby has ability to breathe, the ability to suckle and feed, but it can't do a lot more. And of course, we see this with babies. They're very, limited in terms of their repertoire. But now that child is getting older, sort of learning to talk and walk. By 2 years of age is like a toddler, a terrible toddler who's tearing around and you can't keep track of them. But then that's also reflected in the acquisition of myelin, which is now in the motor areas of the cortex. And now this is a child who's really in a position to learn with fully myelinated fibers throughout the brain. So we only really see fully myelinated brains by about age 2. Again, it can be reflected in behavior. But what about a baby that lacks myelin? So now you can imagine a baby that doesn't have even this. And that's this baby here. This is a baby with Palisades-Merzbacher disease. And what you can see is that this baby actually has a tracheostomy tube, which means it's a tube going into the windpipe to help them breathe, and is also needing oxygen. And so that's sort of a sign of very severe lack of myelin. Even in a baby, they will have trouble breathing, which is almost their only job. But this can also really develop into the later stages of this disease, Palisades-Merzbacher disease. And parents say trapped in a brain without myelin because when you see these patients, they look at you, they're trying to respond to you. You really get a sense that there's a personality there, and yet they cannot crawl, walk, talk. They can't really perform any functions, and sadly, they will die between age 10 and 20 years of age. And this is a disease in which they lack myelin throughout the entire brain. It's due to a mutation of a particular gene called PLP1. Here's what the protein looks like. It's what we call a transmembrane spanning domain protein. It's a protein that's normally in the myelin, but when it's lacking, the myelin doesn't form. And so you have a child that lacks myelin entirely. And this is a child that's probably age 8, who's now in a special wheelchair, probably still needing help breathing, and has a very, very limited ability wouldn't be able to care for itself, obviously, and also has these other problems of PMD. This is a disease of oligodendrocytes. That's also reflected in the MRI. So I mentioned that the normal, 2-year-olds should have myelin. In this image, the myelin appears in a sort of a dark color. So where that blue arrow is, that's a normally myelinated tract, it's dark. But now look at the one on the right. This is a PMD child of the same age, but you don't see the dark color. You see, in this case, a light-looking color. That's not a good sign. That means it's a lacking myelin. So this is a pervasive disease with failure of myelin. So as I mentioned, it has the neurons, it has the astrocytes, the blue cells and the green cells. It actually has the red cells. The red cells can't make myelin, and that explains this devastating impact. Okay, so now let's go to the more of the science question and the translational question. How could we help these patients? Now, there really are no effective therapies existing for these children right now. We try to support them with things like help with breathing and help with feeding, but still, they may not survive past age 20. So let's put on our research hat and let's bring in now the basic scientists and start to think about ways that we could potentially help them. One is, I told you, it's a mutation of a gene called PLP1. Can we fix the gene? I'll talk about that. Could we find a drug that helps the oligodendrocytes from a patient with PMD work better? And I can talk to you about some research that suggests that's possible. And also, could we replace the defective oligodendrocytes by transplanting in new ones? You're familiar with bone marrow transplant, where there may be a failure of the bone marrow the patient has, and then but you can transplant in bone marrow from a donor. Could you do that with oligodendrocytes? And the answer is yes, you can. So let's go back to our building blocks, where I told you oligodendrocytes come from neural stem cells. So you could, in principle, take the neural stem cells, put them in the brain of a boy with PMD, and then would it make myelin? And these are experiments done by a company called Stem Cells Inc. that made neural stem cell product, injected it into the brain of a mouse that doesn't make any myelin. It's kind of a model of the PMD disease. And what you see in the dark color are where the cells have been grafted. So these are cells that have been transplanted in the brain. They've now taken up residence in the brain. They're living there and they're functioning. And with the green stain, we can tell that they're actually making myelin. And this type of evidence would be what we would call proof of concept, that the transplanted cells are able to engraft and they're able to make myelin, and we can check all of that out in the mouse. That's the type of studies that the regulators would then use to say, oh, well, now you can do a clinical trial in humans. And so that proceeded to a clinical trial in which 4 boys with PMD had neural stem cells injected into the white matter tracts. We then followed them over the course of 5 years using MRI and checking them clinically. And we looked at what we would call regions of interest, that in red would be the places that the transplants were put versus another part of the brain that did not receive transplants. And by doing this compare and contrast, we could get evidence that there seemed to be cells that had engrafted in the brain and were producing some of the MRI characteristics of myelin. And that's what's shown in the graphs here on the bottom after a year. These two, subjects 2 and subjects 4, at 4 years after the transplant, also showed changes in the region of transplant that looked like they had living engrafted cells that were producing myelin. And so that gave us further evidence to think that this is a technique that potentially could provide benefit. So what did we find? At 5 years, we found that the procedures were safe. And that's good news because it means that other investigators can now come into the field and potentially do better than we did by providing maybe more cells or better cells than we provided to try to transplant kids with Pelizaeus and Merschbacher disease. We did find that the MRIs were persistently changed, suggesting long-term engraftment. We were not able to prove that they made myelin because these patients have not passed away, and so we haven't actually been able to definitely show that they make myelin. That's a limitation of the study. It didn't really help the children get better because it was too small and it wasn't designed to be able to show that. Another limitation of the study, suggesting we need further research. But fortunately, there are new companies that are getting interested in this idea, and we'll be sort of taking up where we left off because our company that sponsored us went bankrupt before we could do the definitive phase 2 study. So that, just to give you a sense that that is a potential way forward. What about finding a drug that helps their cells work better? Now, for those of you who might be thinking of a career in biology or genetics, we now have this amazing ability to be able to look at the nature of diseases in humans using models that we call induced pluripotent stem cells that can be derived from tissues like the skin. So from a skin biopsy, we can take cells out called fibroblasts. We can use a technique to make them pluripotent. So instead of just making skin, now they can make heart cells, brain cells, liver cells. And that's done through a brilliant cocktail of factors that was defined by Nobel Prize winner Shinji Yamanaka to create induced pluripotent stem cells. What we can also do in the lab is we can take iPS cells from kids with Pelizaeus-Merschbacher disease, and we can correct the gene problem in the dish, and now we have a gene-corrected version. And that means we can compare the mutation effects to one in which we've corrected the mutation, which would be essentially, we think, normal. We can put them through their paces, make them into differentiated cells like oligodendrocytes, and we can even do more. We can even look for drugs that would help those cells work better. So let me just give you a couple of just a couple of slides to show, that we followed this up with our Pelizaeus-Merschbacher disease oligodendrocytes. They would start off as that induced pluripotent stem cell, and then we get them to be a little baby oligodendrocyte. They start to mature into this more bushy looking cell. And then, you'd like them to get here to the myelinating cell, but with the Pelizaeus-Merschbacher disease, they didn't do that. And the skull and crossbones is where what we saw with the cells that had the mutation for PLP1, they died at the pre-myelinating stage. Now, already you can start to say, oh, now we know why they're not myelinated, because what's happening is they get to that stage and for some reason they die, and so they never make it here. And then we see that in the brains of the kids also. They don't make myelin. And now we can also ask more. We can sort of also maybe try to understand this from the point of view of the oligodendrocyte and what this cell has to do when it's not yet myelinating to get to that stage. I like to show this picture because I showed you that it's like a little octopus sending off arms to coat multiple axons. This is a way of showing the output of one oligodendrocyte. Now, this is a tougher job than any of us will have in our entire life, probably. It has hundreds of arms, and it is coding hundreds of axons, and there's one little cell in the middle of that haystack making all of that. And of course, that all has to be synthesized. They increase their size 7,000-fold, massive metabolic stress, and it's right when the cell is being asked to do this that they die in Pelizaeus-Merzbacher disease. So we then wanted to look at what's going on here. We found that they make very toxic metabolites right at the point when they're being asked to take on this really stressful task of growing 7,000 times bigger. And we can then go further and ask what the nature of that toxicity is. And I won't take you through that, but just to say that we found that the cells become very, very sensitive to iron, and iron can be a toxin that can lead to the formation of the toxic metabolites. Fortunately, because we found it was iron, we could also start to think about drugs. And there are drugs that are used to treat iron overload. And those are used commonly in thalassemia patients and others. And so we could use so-called iron chelators to see if that would help the cells work better. So here are the cells that have not been corrected. They're alive, these little red blobs. They're nowhere near as complex or happy as the cells that have had the gene correction. These look like, a healthy octopus. These look like not a very happy octopus. But now if we give them the iron chelators to remove the iron, now they're happy, big, bushy, they survive. And it really did correct the problems that we saw in the culture dish and also in vivo in mice when we gave this iron chelator. So here's an example of a drug that could help the cells work better. The way we would give this in children would be a drug called Feraprox or deferoprone. And so just to take you through what I've told you about these couple of therapies, I've told you about the neural stem cell transplant study, which is safe and warrants further testing, that the PMD oligodendrocytes can be rescued by iron chelation, and now that is going to be taken forward in a clinical trial in Amsterdam where kids with Pelizaeus-Merzbacher disease will go on to deferoprone. There already are some kids that are now taking this, and for what it's worth, the families think it's helping, but we really need to have the clinical study to be able to show that. Okay, and then last but not least, what about fixing the mutant gene? You've probably heard about gene editing, and you've probably heard about all of the great techniques that are now available to think about genetic diseases and different approaches that can be used. I'll talk about one which is becoming really sort of more common in practice, which are called antisense oligonucleotides. And this relies on the biology of how proteins are made. We know that the DNA is the template for proteins. From that, we make mRNAs, and those can be translated into the good proteins that we need in our cell. But they can also, in a disease, be translated into a bad protein that causes problems. In PMD, we'd be talking about a disease-associated protein that is a bad one and is causing that cell death and that iron toxicity. So in this case, there's a good logic for saying we want to just shut this down. We want to turn off the PLP gene and see if that helps. And so now we can use an antisense oligonucleotide that binds to the mRNA. It converts it from that single strand to now it looks like double strand. That can't be translated, no protein, and so it's a logical way to go forward. And this was a study carried out by a colleague, Paul Tazar, at Case Western Reserve University in Cleveland, Ohio, in which they did exactly this. They took a really severe model of PMD. The red survival curve shows that those mice untreated survive only about 3 weeks. And they're not very happy. They have seizures and all the problems of PMD. But now when they're given the antisense oligonucleotide, they have survived for 8 months, which is remarkable, especially for that mouse model. And now that is a study that is also going forward to clinical trials. And if there are pediatric neurologists, you may start to see that now there will be clinical trials for PMD using antisense oligonucleotides. Okay, so how can we help PMD patients? There are 3 potential ways forward, and they're all the subject of active research. One is gene therapy, things like antisense oligonucleotides. Repurposing drugs, the example I gave was deferoprone. And cell-based therapies like neural stem cells or oligodendrocytes that can be transplanted into the brain. And I also want to just end by talking about the relevance in our region, East Anglia, where Cambridge is located, which is a region of about 5.9 million people, and how that is not dissimilar from Singapore, about 5.5 million people in Singapore. But about 5% of our population has a rare disease. So in both our regions, there are about 300,000 people with rare genetic diseases. And so we, as researchers, want to think about the types of therapies, the types of approaches that I've just described that may be ones that we can develop to help patients with rare disorders. So what we've covered, I hope, building a connected brain, the structure and function of myelin, its role in evolution and brain complexity, its relevance for learning, and also the new therapies of the future for rare genetic disorders. With that, I'd like to thank you very much for your attention.